Right. Good afternoon, everyone. Thanks for coming to my talk. I hope you enjoyed your lunch. I'm going to start with a confession. I don't have any photos of cute animals in this talk. So I hope that you can just hang in there, and if you bear with me, you might get to see some cool fungus, okay? Right, so this is our tree of knowledge, and I'm going to use it to tell a story today. That story is about forest structure and forest function. So I asked this question: can we assume that restoration of forest structure will trigger recovery of ecosystem function as well? There is quite a lack of research linking these things in urban contexts, and that's kind of a shame because we then can't do targeted management with our restoration practice. So it's quite an important thing to try to get to the bottom of if we can. So my co-authors and I looked at nutrient cycling as a measure of function. Specifically, we looked at decomposition and denitrification. So no koalas, sorry. We looked at leaf litter decomposition, which is this process by which a leaf falls from a tree, or many leaves, and these subsequent steps that the leaves undergo to be broken down and incorporated into the soil. With denitrification, we see this process by which nitrogen in its mineral form in the soil gets transformed by microbes, so these guys here, into the gaseous form of nitrogen. And this is the only way in which this can happen and the nitrogen cycle can be completed. And this is important, and we should care about it, because we have a lot of nitrogen on our landscapes these days. So these microbes are quite important, and its function is important to have in our ecosystems. It can only occur in hydric or wet soils because it's an anoxic process, and there needs to be a lot of organic matter or carbon in the soil. So why leaf litter decomposition? This is a crucial function to complete nutrient cycling in forests all over the world. So here in Australia and New Zealand, where we did this research, or anywhere else you find a forest. And especially when it pertains, how it pertains to restoration is these systems that we work in are often altered because there's an invasion of non-native species, right? We all work with these. And these non-native species haven't co-evolved with the system they're in, and they can often have different leaf decomposition traits. So we need to understand how this works in with what we're trying to restore. Why denitrification? Okay, well, urban forests have inflated nitrogen levels, as do most other landscapes on Earth today. And this, to bring it into the restoration context again, can affect how well we can do our work, because a lot of our non-native plants are weeds in these systems, and they're very nitrogen hungry. So they get the upper hand if there's a lot of nitrogen in the soil. So there are two main questions I'm going to talk about today. One is restoration of forest structure related to decomposition and denitrification rates at all? And two, what are the key direct and indirect drivers of decomposition and denitrification? If we know this, we can manage for that. So to get at these questions, we use a structural equation modeling approach. And with this approach, you first create an a priori model. You look at the literature that's out there, so you can see that on the left-hand side of my figure here, and you create a structure of ecosystem variables that are interacting together that you posit ultimately are driving the functions you're interested in, or the variables you're interested in. So in this case, denitrification and decomposition. I'm going to point out canopy openness here because this is our metric of forest structure in this study. And this is just how much the forest is filled in its canopy. So we've planted trees, they're growing. When do they actually fill in and create the green ceiling of the forest? Right, okay, so I'm going to describe a little bit about our data collection so that we can test our model and see if those variables actually all interact the way we think that they do. So, as I mentioned, we did this in New Zealand. We measured these 12 ecosystem properties in 2 different cities in the North Island, in Hamilton and in New Plymouth. And we did this across a range of 27 restored forest patches All of them evergreen, native, temperate rainforest. And here's our a priori model again. I do not have time today to go through the methodology for our data collection for all of these variables. Plus, I'm trying to have mercy on your sanity. It's the last day of the conference. So I'm going to describe here the methodology for these two, but you can talk to me afterward if you want to learn about the red box. So for decomposition, we collected fresh, naturally dropped, naturally senesced leaves from the forest floor. These were Welsh mediatala leaves, so this is one of our native trees in this part of New Zealand. And we oven dried these leaves. We didn't collect them from our experimental plots. We stuffed the dried leaves into these bags with dimensions 20 centimeters by 20 centimeters. And we put approximately 5 grams of leaves per each bag. We didn't want to break the leaves and jumpstart decomposition. So about 5 grams, measured exactly how much it was. Each bag was individually identified. We then pegged these to the forest floor, and there are 9 of them at each site. And we did this part of the experiment in Hamilton only. This is what they look like after being out in the field for a while. So you can see the green a little bit, but there's a lot of organic material that's fallen on top of them. That's what we want. We want them to be part of the system. We're measuring how the decomposition is occurring in the system. And after they've been in the field for 14 months, we retrieved them, cleaned the contents of any soil, dried what was left in the bags, and weighed it. And this allowed us to calculate a proportion of mass loss for each bag. And let us see cool things like, here's the fungus I told you about. This is a basket fungus. And this is a weird bag, because this is the only bag that came back weighing more than when we deployed it, because of the basket fungus that was all scrunched up inside of the bag. So that was really cool to see some of the detritivores in action. And actually, these smell terrible, though, if you've ever seen them. Okay, so for denitrification, we did some soil coring. We cored to 10 centimeters depth because that's where these microbes are fully in action in the soil. And we did this 15 times at every forest site and homogenized those soil samples. Then it was back to the lab where we did denitrification enzyme assays. And these they allow you to measure the microbial denitrification potential in the soil. And we did this using the acetylene inhibition method, which is a pretty standard technique. Okay, so that's the how. Now let's answer those questions that I posited in the first place. Let's go up into the boughs of the tree and look at the fruits of our labor. First of all, we decided to investigate some bivariate scatter plots, just between 2 variables at a time. So decomposition is always shown on the y-axis here. And the different ecosystem properties of interest are on the x-axis. And I'm showing you only the significant relationships of all of these, which were 2. In the top left there, both of them positive. Soil moisture and herbaceous plant cover were both significantly positively related with the decomposition, which isn't too surprising. All right, what about denitrification? Here, same layout, denitrification's on the y-axis, and this time there were 4 significant relationships. 2 positive ones, soil moisture and carbon levels in the soil were positively related with denitrification, and this is definitely not a surprise based on the literature. And then pH and sand were negatively related to denitrification. So that's a little breakdown of 2 variables at a time, but what about that big structural equation model? Let's just look at the whole ecosystem together, all the different variables. Here I'm showing you our boiled down final structural equation model. So when you're building a structural equation model, you run a lot of different iterations through, we use R to do this, and it basically tells you which variables are significant and which ones aren't, and which pathways aren't significant. So if you find a variable that's not impacting your system, you remove it. So what's left is all the variables that are significantly interacting in this framework. The arrows are our pathways, and next to them they have standardized path coefficients. So that can range from a value of 0, which is not a good relationship, to 1, which is really strong. Positive arrows are black, and the negative arrows are gray. So the data fit well. We're happy with this overall. But what about our questions? remember that first question? Is restoration of forest ecosystem structure related to decomposition and denitrification rates? We can find out the answer by following the arrows. With canopy openness, which is our measure of forest structure, yeah, there is a connection to decomposition. You can trace the arrows through the pathways. You see there is a significant relationship. However, not so much with denitrification. It's completely independent of that forest structure measure. Instead, it's driven by abiotic properties, and mostly like soil, edaphic properties, topography. Basically, drainage is a big player here. So, what are the direct and indirect drivers of decomposition and denitrification? I've touched on it a little bit, but for decomposition, that herbaceous plant layer and humidity are really key in driving that function. And they're both controlled by canopy openness. So it's all about getting your canopy to close, and then you can control that decomposition level. And then for denitrification, like I said, it's mostly soil properties. Now, I want to point out something interesting to you if you decide that you love structural equation modeling, and this is really interesting. It has some really great ways of pointing out things about ecosystems and maybe your study system that you wouldn't be able to find otherwise. For example, here we can see that canopy openness is significantly related to decomposition. But if you look at it in a bivariate sense, there's no relationship at all. So you would never know that canopy openness was important in this system to get the function that you might want. In addition, you can find opposing effects in your ecosystems. So I've circled here in blue that canopy openness positively affects the herbaceous plant layer, but negatively affects humidity levels. And these are opposing effects. So depending on where your canopy openness level is, you're going to be able to control these two different ecosystem properties in different ways and manage for them in different ways. So this is a really great way that I recommend for you to look at an ecosystem and understand all the different players together. So in summary, is forest structure connected to forest function? For decomposition, yes, it definitely is. It's definitely driven by canopy openness. But for denitrification, no, it's not. It's actually independent of forest structure, and it's driven by the topography and the soil properties in the area. And this implies to me that it is very important to conserve wetlands and low-lying areas, because you can't necessarily build a forest somewhere else to create that function again. So this is a distinction. This isn't a failed lesson. This is actually a really great lesson in knowing where should we put our management energies, where should we conserve, and where can we recreate ecosystem function if we need to. So I hope that whether you're an academic or a policymaker or a practitioner, that this will help you understand a little bit about forests and about restoration, especially in an urban context. And I would like to now acknowledge a lot of people who have helped with this project, and funding and home institutions. And with that, I'll take questions. Thank you.
Thank you, Kiri. We have time for a question or two.
Yeah. Okay. Would you explain the differences between different species of broadleaf, sub-canopy, dropping leaves contribute to the leaf layer? Is tower dominant, or are there other species that can be more waxy or less waxy?
Yeah, so the traits of the species that are in the ecosystem should have an impact on composition. But here we're looking more at the abiotic characteristics of that canopy openness or closure. So that's more about just whatever species are there, how much are they changing light, changing humidity levels, and creating canopy. But yeah, definitely. If you want to dig into it more, you can look at that.
I have a couple of short things I wanted to ask you. I find it really interesting, and of course, with nitrogen, nitrous oxide is something we have to worry about because nitrogen knocks gaseous nitrogen. But there are always variables in this kind of experimental work. Do the taro leaves tend to decompose on the surface, or do they get dragged into the soil by the mesocosmones at all, according to your observations?
Well, ours had stayed in the mesh bags, so those stayed on the surface.
Yeah, no, I mean generally.
Generally, I haven't read anything about what might drag them underground, so I don't know, but I wouldn't be surprised if some of the earthworms maybe introduced
Okay, thank you very much indeed. We stay on the theme of decomposition, this time in relation to tropical forest, and this time from Marisa Stone from Griffith University here in Brisbane.
So the red one works and switch it.
If the speakers are moving around and you can't hear, it sounds like the sound is good, but I can give them this, so just yell, please.
It also helps too because the sessions are being recorded. So if anybody is tuning in on like Echo 360 later, if you guys use a mic, then they can hear you. Yeah.
It's kind of handy if the speakers stay by the screen, really, if you can. Okay. So, Marisa, thank you very much.
Okay, great, thank you. Okay, okay, it's great to speak straight after Kiri because she gives some good background information and to decomposition and also the methodology, which I brush over a bit. So, okay, but this, I'm talking about leaflitter decomposition, and also the role that large invertebrate decomposers play in that process. During restoration on old disused pastures. So worldwide, a significant amount of forest is converted to pasture and many ecological processes are lost. Many of these are subsequently abandoned, so this provides the potential for rainforests or forests to regenerate naturally. However, there can be lasting effects from pasture and some extra help or active restoration may be necessary to help the vegetation along a bit. And one example is assisted natural regeneration, and methods used in this can include removing non-native species to allow natives to regenerate naturally. It's also important to regenerate or recover the ecosystem processes, particularly those that are important for plant recruitment and maintenance, such as leaflitter decomposition, which was explained nicely by Kiri earlier. Okay, so I'll just briefly describe about the process. So litter contains the majority of nutrients necessary for plant growth and maintenance, but the amount that's made available to plants depends on the rate of decomposition. And decomposition, as you heard, is a complex process, and it can depend on a specific set of physical conditions, including moisture, humidity, as well as biological conditions, including invertebrates and bacteria and fungi. They all play a part. So, and of the invertebrates, particularly the large ones are important because they really accelerate the rate of decomposition by consuming and breaking up large amounts of litter, and then this can have a positive effect on the ability of smaller invertebrates and bacteria and fungi to convert the nutrients into a form that plants are able to use. So, for example, large invertebrates, slaters or woodlice, millipedes, amphipods or landhoppers, and earthworms, for example. Several studies have shown that that decomposition rates and invertebrate communities can decline after deforestation, but they can return when the vegetation returns. Increase when the vegetation returns. We know a little bit less about the recovery patterns during this process, though, and this is probably due to the fact that few studies have used both pasture and old growth as benchmarks for comparison, assessed decomposition and invertebrate decomposers simultaneously, or compared different kinds of reforestation approaches. So my key research questions for my research is looking at how deforestation for pasture and subsequent management of regeneration affect leaflitter decomposition and the assemblages of large decomposer invertebrates. My study area is based on the Gold Coast hinterland at Numbamba, where the council has been applying assisted natural regeneration for at least the last 10 years, and they've been very accommodating with me selecting my sites and using the area down there.
So there I have 5 different site types, 5 replicates each, and each of them represent a different stage of recovery. So I have sites in pasture, also have sites, what I'm calling unassisted natural regeneration, which have had cattle removed for at least 5 to 50 or potentially even longer years. And then I have the restoration worked areas, which is the assisted natural regeneration, or ANR, I'll just call it for short from now on. Those that began 1 to 3 years ago, and then sites where it began 5 to 10 years ago. And then, of course, old growth as my reference vegetation. So these sites were selected on areas that were previously cleared rainforest, which then went into pasture, were cleared and went into pasture for 30 or more years, and then have some form of woody regrowth. Except, of course, the pasture sites, which are still in pasture, and the old growth, which were never completely cleared anyway. So to give you an idea what the regenerating sites can typically look like, so this is an example of an unassisted natural regeneration. So you can see there's a big wide open canopy there, and down the bottom here it was just a big sea of the non-native lantana just covering the whole area, smothering everything out from preventing the natives from growing in there. And then if you can imagine when you come in with the restoration methods, if you can imagine the top one where all the lantana has been removed, here all the brown bits is old dead lantana just crushed down to the ground, and then you've got a whole bunch of pioneer tree seedlings have popped up naturally through there. And then if you can imagine that another 5 to 10 years later on, where they've continued to suppress the non-native species, and all these seedlings have grown up into bigger trees now, so you've got a lot of thin stem trees growing. There is a bit more of a canopy forming there, but you can't see that in this photo though. Okay, so to measure the leaf litter decomposition rates, I deployed 200 litter bags across the 25 sites and left them out for 5 and 8 months. And from those, I measured the dry mass loss. So Kiri explained good methods before. Mine was quite similar. So, so this is what it looked like beforehand, and then this is an example of after 8 months. This one, so quite a lot of mass loss has gone. And for my bags, I had 2 treatments. So one was to exclude the large invertebrates, and that was done simply by just the 1 millimetre mesh size of the bag material. And the other treatment was to allow their access, so I cut a bunch of holes on the top and bottom side so that they could get in there. Okay, I'll just say, so what I found in data analysis between the open and closed bags, so the bags that had large invertebrate access had more mass loss, so which suggests that the large invertebrates were contributing to the mass loss. But I also found that by 8 months, or even by 5 months, most of the decomposition had already occurred, so there wasn't a huge difference another 8 months down the track. So I'll just show the more interesting results and just show results from the open bags after 5 months. Okay, so here's a mean and standard error graph of the mass loss percent across the vegetation types, and linear mixed effect models and post-hoc tests revealed that, or confirmed that pasture definitely had the least amount of mass loss, and that old growth up the top here, or forest, had the most amount of mass loss. But then I also found that there was no difference among the 3 regenerating stages here, so the unassisted and then the assisted natural regeneration. Okay, so does deforestation for pasture and subsequent management of regeneration affect leaf litter decomposition rates? So in short, yes, they did. They decreased by almost 50% when forest was converted to pasture, but then they recovered back up to about 83% of forest rates in the regenerating sites. To investigate the invertebrate community, the large decomposers, I extracted them from Leaflitter samples that I grabbed off the surface layer within each of my sites at the 5-month post-vag deployment timing. And I extracted them using a Talgren funnel. So that's where you pop them back in the lab into a funnel here onto mesh with the light and heat source up the top. The invertebrates try to get away, so they go down in the funnel and then get caught down into the collection jar. So, and then from those, I identified them largely to class and order, and then sorted them into functional groups, so whether they were decomposers, predators, herbivores, and then also classed them whether they were large or small invertebrates. So from mean and standard error graph again, this time looking at the large decomposer abundance across the vegetation types. So, and testing confirms that yes, pasture definitely had the least amount of abundance, and that there was no difference among the three regenerating stages. But there was no difference between the regenerating stages or the old growth. Okay, and so does deforestation for pasture and subsequent management of regeneration affect decomposer abundance? So yes, I found some differences again. This time they decreased by 95% when forest was converted to pasture, but they returned to similar numbers as old growth or forest in the regenerating sites. So overall, so when forest was converted to pasture, so both decomposition rates and large decomposer abundance decreased. But during the regeneration stages, decomposition rates had partly recovered, but the large decomposer abundance had recovered. And further, the effects of assisted natural regeneration didn't make a large difference, but that's probably because the invertebrates had actually returned within that first 5 years of cattle removal or stock removal. And to explain where the large decomposers may have recovered the same as old growth, but the decomposition rates may not have recovered fully. That suggests that it's the role that the decomposers are playing. They're there, they are doing the decomposing, but it's probably the other parts of the decomposition process, so the other, physical conditions and biological conditions, so humidity, canopy cover, and a couple of those things that Kiri found in her results are important, and also the bacteria and fungi. So, which suggests that the invertebrates are there doing their role. We just need to wait for the other parts of the decomposition process to recover. And that brings me to the end of my... And just like to acknowledge all of these people here from the Natural Areas Management Unit on the Gold Coast, all the lease and land holders and all my field volunteers. Thank you.
Thank you, Marisa, for a clear presentation. Would anybody like to fire a question this way? Again, this is very close to my own heart, this kind of study, so if I may. Sure. I thought there were a lot of interesting points, but one of them made me think, well, I suppose the history of the pasture management and the forest regeneration makes a huge difference. And a grassland would typically, you mentioned microorganisms, would typically be dominated by bacterial communities and a forest by fungal communities.
Yes.
And, do you have any comment on that at all?
Not really.
So it didn't show in your results, did it?
No, it didn't show my results. I wasn't really looking at the bacterial community. But I guess this difference in recovering rates is a lot higher in the forest than the other side. So the invertebrates are there doing the job, so there's obviously something else that's not quite.
Yeah, the liquidity didn't go away, did they?
No.
But in both of the papers, I also wondered, of course, the litter bag does keep different moisture conditions to general. There are always these variables, aren't there?
Yeah, absolutely.
Yeah. Yes.
So you mentioned the different functional groups of invertebrates,
yeah.
But you only spoke about abundance.
Yeah.
I'm wondering if there were differences in the functional
Yes, okay. I have tested that as well. I just didn't get to the showing the results yet. There wasn't a lot of difference either. I mean, there was a difference between those in pasture than the regenerating stages and old growth, but they were pretty similar as well.
Thank you very much, Marisa. Thank you. Our third presentation is from Sophie Hale. Sophie Hale, yes, and she is from a charitable trust called Motu in Wellington, New Zealand, and our topic is there for you to see, the recovery of functional diversity with restoration.
Thank you, Nick. Good afternoon, everyone. My name is Sophie Hale, and I am a research analyst at Motu Research, based in Wellington, just across the ditch in New Zealand. But today I'll be presenting to you research that I did at the University of Canterbury in Christchurch with my supervisor Jason Tilianakis and Matthias Delling, and we looked at the recovery of functional diversity with restoration. Now, it shouldn't be a surprise to any of you that biodiversity is threatened. We heard from David Lindenmayer that we have 2 billion hectares of degraded land to work with, and thankfully wonderful people such as yourselves are undertaking restoration as a really important tool to try to combat this biodiversity loss. Now, restoration projects often aim to reinstate this biodiversity, but often these projects differ in what they're targeting, as you see on this little clip art here. And it poses the question, how do we even measure biodiversity? Because if we don't know what we're measuring, how can we even know whether we meet the targets that we set, if indeed we did set them? Traditionally, and oftentimes, we measure biodiversity looking at species richness or species abundance. So you're looking at the number and abundance of species in a given area. But given that often we're talking about wanting to restore ecosystems to a point where they're healthy and functioning and delivering ecosystem services that we care about, perhaps looking at just species diversity isn't enough. To take an analogy, imagine you have a toolbox, and in this toolbox you have a range of tools that do different jobs and they contribute different roles to the thing you're trying to do. And that way, the toolbox you see on the right is a lot more helpful to you and a lot more useful compared to the toolbox on the left, where you still have a range of tools, but they're all hammers, so you can only do one thing with them. We can see the same thing in ecosystems. So, looking at the assemblage on the right, if we take the simple analogy that the variety in beaks you see represents the variety of ways in which these birds can contribute to the important ecosystem functions of pollination or dispersal, the assemblage on the right is a lot more useful to you than the one on the left, because you're able to contribute more to this ecosystem function when you have a variety of roles. In this way, there's been a shift in focus to look at functional diversity and looking at traits, as we kind of heard from Daniel Laughlin's wonderful plenary this morning. And when we talk about functional diversity, we're talking about the extent of functional differences of the species in a community. So as I said, functional diversity is really important because it gives us a far better idea of ecosystem function than we can get from just looking at taxonomic-based measures alone. So if we see an increase in functional diversity, that means we're seeing an increase in the diversity of the effects that these species are having on their environment, and also the responses that these species are having to the environment. And this is super important, especially in light of future climate change projections, and we know that our world is changing, so we need to know whether these species are going to be responding in a variety of ways so that they are resilient to this change. And we measure functional diversity using SPATES. Species traits, SPATES, which are characteristics that pertain to the contribution that these species make to ecosystem functions. And when I started this research, there really lacked a comprehensive global assessment of whether restoration works in terms of functional diversity, whether it delivers us something more than just species richness. So that led to our key research question, which was, how does restoration reinstate functional diversity? And part of this is there a relationship in general between species diversity and functional diversity? Because you can imagine that if you are increasing the number of species, you're likely to see an increase in the variety of functions that they hold just by default. So this is something that we wanted to explore further. To do this, I conducted a meta-analysis. So I took 2 recently published meta-analyses at the time I began this work. So Kiri et al. Looked at 108 terrestrial-based restoration projects, and Kyle et al. Looked at 69 freshwater restoration projects. So I read 177 of them, and in order for these to be included in my work, there were 2 key inclusion criteria that were really fundamental when we set up the design of this. And the first one, seemingly obvious, they had to involve intentional restoration. So the studies that simply compared between habitat types or land uses, they weren't enough. There had to be intentional restoration efforts. Now this could be active or passive, and I coarsely categorise these like that, although I totally acknowledge that there are finer details that are important in restoration success. So passive restoration was the removal of the stressor and then the site being left to naturally regenerate, and active restoration was anything beyond that that was more intensive. And what you'll soon see became really important is that we also said the study had to include a negative control. And Marisa demonstrated this beautifully in her sites where she had a pasture site that she was comparing her regenerating sites against. And it means that you can tease apart the effect of your restoration from what might be changing anyway without your efforts. So the sites had to have, the studies, had to have a negative control. In a lot of cases, there was a reference site too, there was a positive control, but this wasn't fundamental and necessary for the study to be included in my work. So this brought it down to just under 50 studies, and after contacting all the authors to get site level species lists, this brought it down to 30 data sets from 28 studies. And these numbers differ because if a study measured multiple taxonomic groups, I split them up and measured them separately. Another important thing to note of these 30 studies is that they differed in study design in about a 50-50 split. So about half of my studies that I looked at measured their data longitudinally. So they went in at multiple points in time and they measured all the sites, the positive controls if they had them and the negative controls in the restored sites. The other half used space for time substitution, which is where you go out at one point in time and you attribute an age to each of your restored sites. And this makes the really important assumption that controls are constant through time. So you're assuming that your degraded sites stay degraded and that your pristine sites can't get any better or worse. And you also assume that restored sites differ only in age, so that there's no effects in terms of spatial distribution or any other regional factors that could play a role. So these are where my data came from. 6 ecoregions and 6 taxa. We looked at just under 1,900 species and about 580 sites. And so those taxonomic groups that you see in the bottom left corner are ants, birds, fish, benthic macroinvertebrates, plants and reptiles. Now, I said earlier that you measure functional diversity using trait data. And to do this, I used a variety of online databases and field guides, amongst other things. But as you can imagine, you can measure wing length for birds, and this tells you an important thing about how it can disperse and what it does, but you can't measure wing length for plants. So in order to, in some way, standardise the trait data that we were collecting, we determined 6 trait classes that were demonstrably important in published literature. So for each of those 1,900 studies that 1,900 species that I mentioned, I gathered trait data for these 6 classes. And I'll say quickly that I won't go into detail and bore you all with my statistical analyses, but I would be happy to talk to any of you about it further. My analyses mainly consisted of mixed effects models and also null model randomizations, so happy to talk about that afterwards. Over to results. So I found that increasing species richness does lead to increases in functional diversity. Now, one of the 3 functional diversity measures I looked at was functional richness, and we saw this positive and significant relationship, but interestingly this curve saturates, which means that In a system where it's relatively depauperate in terms of how many species you have at the start, the contribution that an additional species makes to functional diversity is far greater when you begin with a low number of species than when you're working in a system that is already relatively full. Further to this, in the space for time data, we saw that restored sites did improve over time, which is great. Interestingly though, in the longitudinal data, which is where we could actually measure changes over time in the positive controls, negative controls, and restored sites, we did see improvements in restored sites over time, but we saw improvements in all sites, including the degraded sites, which, as you will see, so the effects differ by study design. You would have heard me say earlier that in space for time substitution, we assume that degraded sites stay degraded and that they cannot change over time. But our better data, the longitudinal studies, we found that degraded sites did improve over time, which is problematic and also really interesting, and it suggests that the assumption that we make in space for time data that these degraded sites don't change may be a false assumption, and in fact that the positive results we see with our space for time data may be an artifact of the very assumption that we made when we set up the study design in the first place. As well as this, the fact that we saw such a strong, positive and significant relationship between species diversity and functional diversity suggests that projects that do look at species richness do tell you a little bit about functional richness too, but this saturates at higher levels of biodiversity. And this is really important if we're talking about something like biodiversity offsetting, which if you have an area that is okay and we are destroying it and we're justifying that by taking a different area that is similarly okay and making it better somewhere else, the cost of taking something from okay to worse is far greater than the benefit you get from taking something from okay to better because of that saturating relationship. In terms of recommendations for future restoration projects, we heard in David Lindenmeyer's plenary that it's absolutely critical that part of your restoration project budget goes towards monitoring. And I couldn't agree more, and from the results of my study, I would stress that negative controls must be a part of that monitoring. You have to have something that you can compare your restored sites against, otherwise you could go back in 5 years, see improvement, attribute it to your efforts, base future studies on that effort, whereas the whole thing was just done regardless of your efforts because of this positive recovery of the degraded sites anyway. And as I said before, I would suggest being strategic. So there's far more to gain in preventing sites from getting worse than there is taking ones that are already good to being even better. And I'd like to thank massively my amazing supervisor, Jason, and everyone from Canterbury and from other affiliations that have helped me get to here. Thank you all so much.
Thank you, Sophie, for a very clear presentation. It's so important, functionality. But I know aged professors who've spent their career trying to demonstrate this experimentally. So I always think it's a brave topic to take on.
Thank you.
Yeah, I have a question. It's really interesting. I'm just trying to wrap my head around the differences. So you said it makes sense the difference in improvements are greater for sites starting with lower species diversity. I was just wondering, just for the sake of understanding, if that affects the result for a degraded site, because it's going to possibly, maybe in comparison, read as a greater improvement on the same chart because it's starting with such a low amount.
It's a really interesting point, and I hadn't thought of it, and that's great. Yeah.
Sorry, maybe I should catch it later. But I'm just wondering if at the tail end of that, at the point of end time,
Sure.
whatever, maybe look at the raw data again, because where for a restored site you might have an increase of 30 species, perhaps for the same degraded site, you've only increased 3 species, but it's reading as a similarly positive trend.
Yeah.
Maybe.
Yeah. Yeah, thank you.
So Sophie told me before the talk it was her first conference presentation. and she had the perfect answer to the... That's very interesting. Thank you very much, Sophie. Next we have James Walsh, who's going to talk about grassy woodlands, Sheoak Grassy Woodlands in South Australia.
The rest looks like it's all online. Yeah.
Good afternoon, everyone. My name is James Walsh. I'm working with Lilter, Australia, and today I'll be presenting a drooping Sheoak restoration project we delivered in South Australia. The drooping Sheoak woodland in South Australia is highly degraded. It's due to a combination of factors, namely overclearing and overgrazing. So when you're driving through the natural range of this particular ecosystem, a lot of the landscape looks a lot like a moonscape, with the sheet limestone is basically all that's left, and the sun's glistening on it, so it's quite surreal. So that's why we decided to scope up and deliver this project. So it's located, the project itself is located on Dakalanta Wildlife Sanctuary. It's owned by Australian Wildlife Conservancy. So Landcare Australia and AWC came together to scope up this project, and basically Dakalanta is set amongst a mosaic of grazing and cropping enterprises. So it's quite strategic in terms of connectivity within the region. This is the red quadrangles there, that's the actual property, so 13,000 square kilometres, 30,000 hectares. And it's also part of the Wild Eyre BioLink program, which is this other highlighted boundary you can see. So a bit of background. The project is part of the 20 Million Trees program, so it was successful in getting funding from the Australian government for that. And that money basically flowed through the National Landcare program. So this is, the project goals were deliver 1,080 hectares of drooping Sheoak restoration and 110 hectares of mallee box or eucalyptus Marisa woodland. And it was also designed to improve habitat for the hairy-nosed wombat, which is a resident on the sanctuary and one of the reasons why it was purchased in the first place. So you can see how tough a site it is. The limestone's evident, and you can see a lot of young drooping Sheoaks emerging there from the seeding effort. So prior to kicking off the project, Landcare Australia set about mapping all the key stakeholders in the region. And once we'd identified the key players, we formed a steering committee. So this included Wild Eyre, EPNRM, AWC and ourselves. And then this steering committee oversaw the delivery of the project, timing of on-ground works, and really helped us access local knowledge in the region. Now the work order itself, rolling out 1,200 hectares of re-veg on very tough terrain, required broad collaboration. So we came together with a lot of like-minded organisations and basically worked together to deliver this project. So all these organisations contributed to the project in some way. We had Trees for Life, GA, or Breeding Australia, and ourselves doing the direct seeding. We had the Sejuna Aboriginal Corporation, Native Seeds and GA also doing our seed collection. And then Wild Eyre contributed a direct seeding unit, and we also had a direct seeding machine specially constructed for the project. So this gives you a bit of an idea of the scale of the project. This landscape's been destocked for quite a while, so it had, in effect, no ability to regenerate naturally, so human intervention was required. All up, we did 2,380 kilometres of direct seeding, and this is across really tough terrain, as I mentioned. You can see some of the limestone in this image. Traditional disc seeders just don't function in this country. They'll get through 10, 20 k's and then they're kaput. So we reckon, so the seeding rate is 5 k's an hour. So in order to optimise seasonal conditions, we needed all hands on deck, basically. So this is our seeding machine we had constructed. The front tine delivers the slow release fertiliser and the large seed, and then we have the fine seed coming out the second tine. So basically when this seeding machine hits limestone, it kicks back and just skids along the top and then comes back down, and the press wheel spins the drive shaft there, which turns the seed boxes. So this is a drone still shot of the landscape. You can see how much limestone there is on the property and how bare it is and devoid it is of natural vegetation. So we separate the large and the fine seed to optimise seed placement. So we work on a formula of 1 kilo of pure seed per hectare, so 60% large seed, so your acacias, your dodonias, your pittosporums, your sheoak, and then your fine seeds, your eucalyptus, your melaleucas, and your leptospermums. So we come in, we do our seeding pass, and then we follow up with the herbicide application, which knocks out the weed layer in the first instance, and we also use the insecticide application to take out red-legged earth mite. We use a penetrant as well. We use pulse in the herbicide mix to make sure we get a good knockdown. Once we had emergence, like germination, we came in with a shielded spray. These are a couple of modified quad bikes with a shield. So basically you drive along your seeding line and the shield, you've got spray jets on an angle that knock out your secondary weed germination. These were developed by Trees for Life SA, so that's their intellectual property, but a great bit of gear for increasing your recruitment. And also, as I mentioned, we also included a slow release fertiliser drop. So based on that formula, it's no small task, but basically we collected 1.2 tonne of locally sourced seed. That's the Sejuna guys processing some Eucalyptus porosa, and this is some of the large seed mix. So you've got your sheoak at the bottom there, Templetonias, pittosporums, very diverse mix of plant species. We also did some climate adjusted modelling, and that indicated that we should collect some of our seed stocks from further north. Because under the modelling, basically, Dakalanta was going to become more dry in the face of climate change. And so some of our seed stock came from further north where it's drier. In total, we used 50 local plant species across 3 distinct vegetation associations. And basically, that also gives us a very robust adaptation mechanism in the face of climate change, just through species diversity. Now, rolling out revegetation projects of any size, the success of any projects depended on good pest management, and we recognised this early. So basically, we identified any water point within 5 kilometres of the project site, and we shut it down basically with working with local farmers. So the compensation and the conversations and the negotiations around that basically involved us providing some revegetation for those farmers, some direct seeding of fodder crops such as saltbush to improve their productivity, and also some fencing materials. So that helped reduce the overall abundance of herbivores on the project site, which was a great start. And then when it came to pest species on site, such as rabbits, snails and mice, we basically surveyed the property and identified wherever there was activity, we GPS'd it, gathered data, and then we designed our baiting program accordingly. This minimised off-target damage and also helped just knock out any pests that could damage the project's success. We also had spotlight controls, and there's some stats on what was removed from the property during those controls. So just monitoring of the project, these are all GPS points of our transects. The yellow points are from 2016 seeding and the red points are from 2017. We did counts every 3 months and we had photo points taken across each monitoring site. So each transect samples an area of 33 hectares. So this is a transect. You can see the before and after, really good recruitment along the seeding lines there. So basically, based on that monitoring program, each transect, I should just give you a rundown, 100 metres sample, and then that's spread across 5 seeding rows, so basically we get a good snapshot of what is happening in that area. The monitoring data indicates that we have 2.1 million plants on site currently. And these are just some more seeding shots. You can see how tough a site it is, but really great results in terms of a landscape that was pretty much, grazed to death. So again, just touching on the formula, 1 kilo of seed per hectare, and some more strong recruitment, across the seeding lines. Really great result for Dakalanta and for biodiversity conservation. So just some points of interest. It's a great project in terms of the collaboration that went on amongst all the different NGOs, government, private sectors, traditional owners. The slow release fertiliser is something that's not usually done in SA when we're doing native veg, but it seemed to benefit the majority of species, and taking a regional approach to the pest management side of things with shutting down water points, really beneficial in reducing herbivore abundance. This is some seed viability testing we did in partnership with the Botanical Gardens in Adelaide. And so just to summarise, the project target was 595,000 plants, so we've exceeded that by 1.5 million. It's been really successful, and basically just under 2,000 odd kilometres of seeding, so a really good news story, and glad to share it with you all. So, any questions?
James, what's 1.5 million for 2 pounds?
Two questions. You said it's integrated to the eye to define seeds. That machine is open in single row, and you sow them in alternate rows. How does the delivery actually work from the small and large seeds?
So there's 2 times in the seeding shot. Probably the front time runs the deeper, and then the back time, fine seed just comes out on the surface, and then the press wheel pushes that in. So in order to optimise the seed placement for the large seed, you want to have about 30 mil, and then your fine seed can just go on top and get pressed in with the press wheel, and you still get, as long as you're sowing into good moisture.
Yeah, but it's the same row, but slightly offset within that one row.
All the seed goes into one priori. Sorry, Paddy?
Yeah, James, what are you using to monitor the plant regrowth?
So with those transects, we just do stem counts along each every 3 months. So basically, we count according to species. So however many acacias there are, sheoaks, and the photo points to really give you the growth monitoring results. So basically we just break it down to how many plants we have in the transect according to each species, and then we let the photos tell the story for the growth rates.
And I was just curious, I don't actually know the system, I don't know what the dispersal mechanisms for the different plants are, but presumably a lot of them only wind dispersion, but I was wondering if there's any fruit-based seeds and what the process was for including that in your sowing, like taking the seed out of the fruit or shortcutting that, or...
Yeah, sure. So, like with Pittosporum, for instance, that's a very waxy cover on it. So you process it before you put it in the ground, because otherwise it'll just clump together. So you have to take all that waxy inhibitor off, and you can do that by soaking it in dynamic lifter. But the seed treatment mechanisms are all done prior to seeding. So all the acacias are scarified and or heat treated, and same with the Dogonias, and then so basically we treat everything prior to putting it in the ground.
Thank you very much. It's very good to see these agricultural technologies being used by people these days. So thank you very much indeed.
All right, thank you.
least, we have Hannah Fraser from the University of Melbourne, who's going to talk about eucalyptus woodland recovery.
Hi, thanks for staying till the end of the session. I'm going to tell you a bit about some research I've been doing with the with NESP, Threatened Species Recovery Hub, which is to do with trying to build a generalised recovery plan for eucalypt woodlands. Okay, so that's obviously a reasonably lofty ambition, and we have to cut the scope down a little bit. So instead of including all of the eucalypt woodlands in Australia, we've cut it down to the southern ones, so starting from sort of the bottom of Queensland and wrapping around through to Western Australia. And we're looking at 3 distinct types of woodlands to be in line with the CSIRO's Australian Ecosystems Model Framework. So that's floodplain and riparian woodlands, grassy woodlands, and shrubby and obligate cedar woodlands. And the problem is that there's 28 EPBC listed threatened woodlands, but only 9 of them have recovery plans. So there's 19 threatened woodlands out there where essentially there's no official guidance on how to manage them. And the question is whether we can efficiently create those extra 19 plans, or whether we really need to go one woodland at a time, in which case probably we're never going to get to the end of all of them. So the overall objective is to ensure that threatened woodlands all have recovery plans where necessary, though I should note that some of the EPBC listed threatened woodlands don't specify that they need a recovery plan, but in my opinion, more advice is helpful rather than less. For the specific project, we aim to build a state and transition model which describes a range of woodland condition states and also the management actions that can drive the transitions between those states, as well as also the environmental factors that might inhibit or encourage those kinds of transitions. And then use that as a basis for a recovery plan which will describe how to manage eucalypt woodlands in southern Australia. So why state and transition models? This is sort of an example of a state and transition model on the left here, where we've divided it into a number of different condition states. And this is an old model that sort of formed the prototype for the model that we built together using some expert data. So we've got different states such as reference, exotic pasture and native pasture, and we've also got management drivers that drive the differences between those things. So native pasture can go to exotic pasture if you fertilise it and sow it with introduced seed, for example. And this kind of structure can help articulate management objectives. So you might be able to say, all right, we've got 90% of our landscape is in native pasture state. Where can we go from there according to this state and transition model, and how do we do it? So it provides both help articulating management actions, and also by understanding the processes behind those transitions, it provides guidance for how to actually achieve the management objectives you've got. So I'm going to outline the steps we took in this process. The ones in black here are the ones we've already done, and the grey ones are ones we're hopefully going to finish soon. So we started by getting together some woodland experts from across southern Australia and asking them to develop qualitative descriptions of these condition states. Then we sent them out a survey and asked them which of these states could transition to which of the other states. So could native pasture go to directly back to the reference state, or does it have to go via a different state on the way? Next, we got everybody back in a room together and broke them into groups for the 3 different woodland types, so grassy, riparian, floodplain, and shrubby obligate, and asked them to specify how those transitions happen. This gave us really good information to start building that state and transition model. The next steps are to turn that qualitative description of the states into something quantitative, which you can use to identify which state you're in when you're on the ground, and then compile the state and transition model and write up the recovery plan. So, based on the qualitative descriptions of the different woodland condition states, Megan Good helped us draw up this great schematic. So along the top here, we've got increasing structural change from the reference state, which is to do with changes in the overstory cover and arrangement. And downwards, we've got increasing change in the mid and understory. So we've got decreasing native plant diversity and increasing exotic cover. And you can see we've identified 7 different condition states, going from reference, which is pretty much what you'd think, to simplified 1, which is sort of like the travelling stock reserves, where it's pretty good, but it's been damaged a little bit through some light grazing and simplification of the mid and understory. Simplified 2's got 2 different variants. It's either got shrub encroachment, or the understory is depauperate, caused by probably grazing again, but at a higher level. Simplified 3 is sort of a native pasture, so it's grazing land, but no, it hasn't been fertilised or seeded with exotics, and it's maybe got a couple of scattered pasture trees left. Simplified 4 is a rare one. It sort of occurs along in cemeteries or along rail reserves, where it would have been originally a woodland, but it's essentially a derived native grassland because the trees have been trimmed out. And then there's thicket, which is super dense vegetation caused often by removing grazing and then having a mass recruitment event. And then we've got an understory and shrub thicket, which is caused by, let's say, slightly over-enthusiastic restoration, followed by good conditions, and ends up not exactly how we intended it. And then transformed is sort of a grab bag of any kind of completely transformed woodland, which essentially doesn't look like a woodland at all. So it might be salinized, it might be a exotic pasture, it might be cropland. So from there, we got people to specify, as I said, which states could transition to which other states. And in this image, we've got different colours for the different types of woodland, and the size of the bar corresponds to how likely those experts thought that transition was. So on the top, we've got the state you start in, and then down the side, that's the state you transition into. So for example, people thought that if you started in simplified 1 state, you could get to exemplar, and you could get to simplified 2. That was true for all of the groups. The floodplains group didn't think it was likely for you to go from simplified 1 to transformed. And that sort of gives you an idea of where the most of the transitions are. So the more colour there is in a square, the more people agreed that that transition was likely to happen. Yes, so the causal transition chains. This is the bit I'm most excited about, and the bit that I'm least far along in analysing because it's very complicated. So essentially we got people to draw this kind of chains where you start with what your starting state and what's the state you think you can transition to, and then you specify the different environmental and management factors, drivers, that you need to get from that state to the other state, and then what attributes you would check to see whether that state transition had occurred. So for example, you might say, I start off in this simplified 4 state, which was that derived native grassland. Then you remove the high intensity of grazing, you get above average rainfall and a large recruitment event, and you turn into an overstory thicket, which you'd know about because of a high immature stem density. I haven't got all the way through analysing this data yet, so I thought I'd just throw up the most common environmental and management drivers that came out of this, so that you can fact check whether this kind of approach came up with sensible things. So management drivers came up that were driving, I'll call them negative transitions, though the intention is not to have this to be particularly value-laden. Clearing trees and having continuous grazing often resulted in what I would consider a decline in the condition of the community, so transitions more towards your heavily grazed, poor understory conditions. Then nutrient removal, revegetation of the understory, weed control, removing grazing, and thinning of the overstory were things that were often came up of ways to improve the condition of the vegetation. Similarly, which links into Valerie's talk from yesterday, I think it was, there's environmental drivers that can either facilitate or inhibit these transitions. So drought, availability of exotic propagules, and soil biophysical processes being disrupted can mean that if you do management actions that would otherwise be successful, you might not actually achieve your success. On the other hand, dieback or self-thinning, if you're in a thicket, can be a good thing. natural recruitment, adequate rainfall, and sometimes flood for the floodplain woodlands can facilitate the kinds of transitions that might otherwise not be possible. So, once I managed to finally compile all of these transitions together and draw up all of the different causal chains behind all those transitions, the next steps are to quantify the different states so that we can identify in the landscapes and compare whether these look different across different regions of Australia and between the different woodland types. Then I'll evaluate the differences between the causal chains according to different woodland types. So, for example, is flooding more influential in floodplain woodlands than in grassy woodlands? I think it probably is, but there'll be more subtle things than that to look for. And then develop a state and transition model that captures all of this information and can be used to inform a recovery plan. And the thing I haven't talked about at all, obviously, is fauna. I've obviously got a vegetation-focused approach to this, but a recovery plan really needs to include fauna as well. So we're hoping to do some extra workshops with fauna experts to try and integrate them into this model, but watch this space for that. It's in its infancy. I'd like to thank my collaborators and the great people at the Department of Energy, the Environment and Energy for facilitating this project.